Good evening, I'm Felicia Ruiz, and this is my Module 4 video submission on the process of COVID-19 RT-PCR testing video. So I heard that you're nervous about how you test positive for COVID with a COVID swab test, but my husband's a lab tech and he actually explained to me how the process goes. So it is a very common lab technique. RT stands for reverse transcription and PCR stands for polymerase chain reaction. And basically it's used to copy segments of the DNA, and in this case, it is used to copy and pull the virus as the genetic material. So positive means that you have the SARS-CoV-2 virus, and negative means that either none was detected or there's too little of the virus, of the genetic material. And so what they're going to do is they're going to either swab you nasally on the inside of your nostril, and they get pretty high up there, so it might be kind of uncomfortable. And they put that swab into transport media or into normal saline. And then they place that sample, they pipette that sample into these injection vials. So the PRC machine, there's multiple versions. A common one is the BioFire. You put the vials in there and you pipette the sample specimen. And then once they're in the machine, you start to run the machine. It takes about an hour to actually do the whole testing process. But once you do that, the PCR is converted from a single RNA strand to a double DNA in this process of reverse transcription. So the two DNA strands are then separated, and then the primers attached to the ends of the strands and to specific sequences or specific segments of the virus. And when it attaches to those viruses, that's when the fluorescent dye comes in. And you get these new strands of DNA that extend off. And then this cycle gets repeated dozens of times, probably like 30 or 40 times in the process. And so when you do get a positive result, the results will be millions of copies of these primers with the matching DNA on the results. And then if you don't, there's not going to be any. There's not going to be any of these. It's not going to have multiple segments of the dye. And that is the PCR process into testing for COVID virus. Thank you.